Today, in part 2, I would like to explain the overall picture of the mechanism of coronavirus infection and introduce the contents of research on therapeutic targets and international joint projects related to COVID-19, which is being developed in OECD, etc. First of all, this is the mechanism of the infection of coronavirus. Virus particles inhaled from the nose and mouth reach the lungs through the airways and infect the cells. The virus amplifies in human cells, causing inflammation in the lungs and the whole body and cytokine storms, which are excessive immune responses, and becoming severe disease. A recent paper in Cell has been studying the differences between COVID-19 and common respiratory infections, and according to this paper, the induction of interferon 1 and 3 is weaker than that of common viruses in SARS coronavirus 2, the virus of COVID-19. Instead, it was found that SARS coronavirus 2 strongly induces chemokine and relatively strongly induces interleukin 6, etc., which causes inflammation. This paper was published in Nature Nanotechnology, but the mechanisms and targets for coronavirus infection in cells are well summarized. First, virus particles bind to H2 on the human cell membrane, blocking this binding with antibody drugs and serum therapy. After that, some proteins are proteolyzed by protease called TMPRSS2, and viruses are encytosed in the cells. And the inhibition of this protease is also a therapeutic target. After endocytosis, the viral particles release the viral genome and are amplified by RNA polymerase. This inhibitor of RNA polymerase is remdesivir, the only approved drug in Japan as of August. After this, the viral genome is put in the virus capsule synthesized in the human cell. It is released into the extracellular space and the infection spreads. This is a schematic diagram of the genome, genes, and proteins of various coronaviruses in a paper published in Nature Review Microbiology in 2019. Coronaviruses are broadly classified into alpha, beta, gamma, and delta. The SARS coronavirus 2 is classified as a beta coronavirus. In the beta coronavirus, SARS coronavirus, MERS coronavirus, and the SARS coronavirus 2 are present. There are therapeutic targets for the three coronaviruses proteins synthesized from their genome. These treatments for SARS coronavirus 2 include nafamostat targeting S proteins and remdesivir targeting RNA-dependent RNA polymerase, which I mentioned in previous slides. Tocilizumab targeting interleukin 6 receptors involved in immune responses has also been developed. This is an introduction to the research of the target candidate for the COVID-19 treatment. When we analyzed the upstream regulators of coronavirus pathogenesis-related molecules introduced in the movie, or part 1, immunosuppressive agents and prostaglandins, etc., were identified. The network of prostaglandins is like this, and the interaction with existing drugs labeled in orange and with microRNAs labeled in pink and were analyzed. I would like to introduce the AOP program currently under development in OECD, etc. This is a project in which countries cooperate and develop adverse outcome pathway AOP for cellular responses and biological effects caused by various chemicals and molecules. We are developing signaling pathways by environmental substances, therapeutic agents, and various other triggers internationally. Currently, a project to develop a COVID-19 AOP called CHAO is now in the process of being launched. After this movie, I hope you will take a look at this CHAO introduction video.